Thank you, Victoria, and thank you to the Stanford Blood Center for inviting me to come share our research that we do in the Clegg Lab at UC Santa Barbara. So my talk today is about personalized medicine using skin and blood cells to cure blindness. And first I'm going to go over how I'm using that term personalized medicine, because it's going to be a little different than how it's traditionally talked about. And then I'll go over how we take skin or blood cells from patients and then use that to learn a little bit more about their specific blinding diseases. So traditionally, when you think of medicine, you think of some group of patients with a disease, and the doctor prescribes, usually, the same drug for that whole group of patients. However, doctors have known since the age of Hippocrates that there are inherent differences in these individuals that all have that same disease that are going to affect how they react to the medicine that they're given. And with the onset of DNA testing and more of that data becoming available to patients and their doctors, scientists and doctors have tried to figure out how they can use that genetic information about these patients to better treat them. So in some cases, doctors have started to look at that genetic information and either give a lower or higher dose of that medicine, or maybe an entirely different medicine to that patient. So the form of medicine that my research focuses on in the Clegg Lab is actually an entirely different modality. And that is, ooh, did my batteries die? Oh, no, we're good, okay. Stem cell research. So imagine this group of patients here all have a disease that they need neurons for, as pictured in this cartoon schematic. And what we want to know for stem cell therapies is how we're ever going to make this a medicine. What are the obstacles to getting cell therapy to be truly a medicine available to the public? And the first would be that researchers need to be able to understand what cell type it is that the patient needs, whether that be neurons for something like Alzheimer's or Parkinson's, a pancreatic cell for diabetes patients, or an eye cell for blind patients. And secondly, doctors need a way to surgically transplant those cells into the patient. And then finally, how are we going to tailor these cell treatments to individual patients in the same way that doctors are trying to tailor drug therapies to their individual patients? So what is it that we need to know in terms of genetic information, et cetera, to make sure that our cell therapies will work for all of the patients with a given disease? So that's my main question for the talk today, is how do we apply the concept of personalized medicine to stem cell therapies? First of all, where do stem cells even come from? So back in 1998 was the first time that scientists were ever able to culture stem cells in the lab. And they were first isolated from excess embryos that are produced during in vitro fertilization. So in this process pictured here, as the fertilized embryo develops in the lab, when it gets to the day 5 stage, those cells can be removed and grown in lab. And what's really the two defining characteristics of these cells is that one, they can divide indefinitely and self-renew, producing infinitely more stem cells. And second, they can produce all 3 germ layers of your body, meaning that they can produce all of the cells in your body. And so this work was pioneered by Dr. Jamie Thompson. He's out of the University of Madison, Wisconsin, and was actually featured on the cover of Time for this work. He also has a partial appointment at UC Santa Barbara, so we've been lucky enough to work with him in his lab on his amazing stem cell technologies. So in addition to embryonic stem cells, there's actually a few other sources of stem cells that maybe you haven't heard about. So this image here is called Waddington's landscape. And basically, it's a really great metaphor for how these cells develop. So this black and white ball at the top here is the fertilized egg that I was just telling you about. And those cells are what are called totipotent, meaning that they have the potential to become all the cell types in your body. But the natural progression of this is for the ball to metaphorically roll down this hill and divide and become different types of cells in your body. So they go through a state of being pluripotent, meaning they become most cell types, or something that might be multipotent, something that can become many different cell types, and then finally go on to become all of the different cell types in your body, including things like skin and blood cells. And interestingly, some research focuses on those adult stem cells for therapy, so the most famous of which would be a bone marrow transplant, and that actually relies on stem cells that exist naturally in your bone marrow to replenish all of the blood in your body. But the cells that we work with are actually entirely different from those embryonic stem cells and these adult stem cells. So a while ago, scientists were wondering if you could reverse this process. So as I said, cells naturally go from a state of being more potent to more differentiated. And we wanted to know if you could reverse this process, if you could give these cells some form of amnesia and force them back to a state of pluripotency. And this amazing reversal is now possible because of a technology that was invented back in 2007 called Induced pluripotent stem cells. And so the way this works is when you have a donor or a patient, you can take a skin sample from them in the form of a biopsy. And those cells, called fibroblast cells, can be exposed to 4 what are called transcription factors. And what transcription factors do is travel into your cell's nucleus, where all your DNA is, and it tells the cell to go back to being a stem cell, to go back to being something that can self-renew indefinitely. So with this work, they showed that they could produce what they now call these induced pluripotent stem cells that can self-renew just like an embryonic stem cell, but without any of the ethical concerns. And so from here, there's some really exciting options for researchers and doctors. The first of which is that these stem cells can become any cell type in your body, and many different labs have produced ways to make muscle cells, neurons, heart cells, so on and so forth. And you can imagine with something like that, you could take these cells through a drug screening process where you grow this patient's own cells in lab and you screen for different drugs to see how effective they are or maybe potentially toxic they are to that specific patient, really elevating this concept of precision or personalized medicine. Another route to take is to genetically edit these cells, which I'll return to since it's a bit of a hot topic these days. And what you can do with these corrected cells is then look at the difference in lab between the original disease cells and then the genetically edited cells and try to figure out on a cellular or molecular level what's going on for that specific patient that donated those cells. And then finally, the ideal option would be to take those corrected cells and transplant them back into the patient with the disease. And not only are we working on skin cells, but more recently, scientists have tried to figure out how they could take a blood sample from a patient, like they do here at the blood center, and take out a certain fraction of cells that still have their nucleus. So red blood cells don't have a nucleus, but there are those white blood cells in your blood that do have a nucleus and can go through this same sort of reprogramming to become stem cells. And the obvious advantage there is that that's a much less invasive way to get cells from a patient than a skin biopsy, which can be a little bit invasive and is a small surgical incision. So what's really interesting about this story too is that there were actually 2 labs that published this work on the same day in 2007. So Jamie Thompson's group published it in Science magazine, and a man named Shinya Yamanaka and his group out of the Riken Institute produced the same result, showing that these induced pluripotent stem cells can become all 3 germ layers of the body, just like the embryonic stem cells, again, without any of those ethical concerns. And what's so exciting about this is that Shinya Yamanaka actually went on to get a Nobel Prize in Medicine in 2012, really demonstrating how important this discovery was to the field of medicine, both regenerative and personalized medicine. So with this amazing technology now available to us, there's a multitude of different disease models that you can study. And what we focus on in the Clegg lab at UCSB is the eye. So this is a cross-section of an eye with the retina blown up in more detail on the right here. And as light passes through your pupil and hits the retina, the back of your eye, it comes into contact with these cells called photoreceptors. So these are light-sensing cells, rods and cones, that can convert a light signal into a neural signal, and it travels back your optic nerve and into the back of your brain to form an image. Maybe a less known cell type in your eye are these supporting cells called retinal pigment epithelium. And that's this monolayer that sits at the back of your eye. It's an epithelium just like your skin is, where it forms this nice barrier layer. And you actually don't need a microscope to see these cells. If you stare into your neighbor's eyes, you will notice that their pupil appears black. And that's not because it's dark in there. That's actually because this layer of cells is so highly pigmented in order to absorb all that stray light that travels into your eye and really help keep those photoreceptors healthy. So you can imagine a disease that affects either one of these cell types will eventually lead to blindness. And briefly, I just want to mention, why are we studying the eye as a starting point for understanding stem cell therapies? So one of the main reasons, other than the fact that the eye is a beautiful and complex organ to study, is that there are already advanced surgical methods that are in place for the eye. There are ways that surgeons can manipulate the back of the retina. So in terms of using stem cells in the back of the eye, that is actually already in progress. And then there are good endpoint parameters. So with stem cell therapies, you really want a quantitative way that you can measure that the stem cell therapy affected the patient or improved the patient. So with vision, there's good ways that they can actually measure the electrical activity of the back of the eye after the transplant has occurred. And also the eye is immune privileged, which can help a little bit with concerns in terms of immune rejection. Anytime there's an organ transplant or a stem cell therapy, there's always a risk of immune rejection. And I say possible immune privilege because, of course, in a disease state where the retina and your eye has started to deteriorate, you may have lost some of that immune privilege. So the two diseases that we study in lab are called age-related macular degeneration and retinitis pigmentosa. And these images here are showing you what the patients experience. So normal vision on the left, compared with losing the central field of vision in macular degeneration versus this sort of tunnel vision that happens in retinitis pigmentosa. And for macular degeneration, as the name implies, this is something that comes on with age and is not necessarily linked to one gene. However, retinitis pigmentosa comes on in children and young teenagers, starts as a form of night blindness, and then the periphery of their eye and their vision starts to deteriorate, and by middle age, some of these patients completely lose their vision. And these images here are fundus photographs. So this on the left is a healthy retina. That bright yellow spot is where the optic nerve is, and that darker red spot is your macula, where sort of all your fine focusing happens. And you can see in age-related macular degeneration, you're getting these yellow deposits built up, and it's destroying the vision in the macula and then slowly progresses to the rest of the eye. And interestingly, my boss, Dr. Clegg, and our work with those pigmented cells are now in a clinical trial for this disease, trying to use that stem cell therapy specifically for this disease. And then for retinitis pigmentosa, you get this signature phenotype of this kind of hyperpigmentation creeping in from the periphery of the eye and slowly leading to that tunnel vision. And just to give you an even closer look at what that looks like in terms of the morphology of the eye. You have a normal retina on the left here where those rods and cones are nice and aligned, and then a patient with retinitis pigmentosa where you can see that the retina is severely degenerated. So what's not known about this disease is how mutations in certain genes lead to this phenotype. And so for the rest of the talk, I want to focus on how we're using these IPS disease models to get at this question of what's happening here on more of a molecular level. And returning to this concept of induced pluripotent stem cells, I'd like to mention why we even bother making stem cells at all. Why not go into the patient and just isolate some of the cells that we're interested in studying and trying to understand them directly, like the photoreceptors? One of the more obvious reasons, as I previously stated, is that you want to be able to get cells from a patient in the least invasive way possible. And a skin biopsy was a great start, and blood samples are an improvement upon that. And secondly, like I said, a really defining feature of stem cells is that they can self-renew indefinitely, meaning we can produce millions of these cells in lab to experiment with and have countless cells to work with. However, with things like even the skin cells or the blood cells or photoreceptors, they have a finite lifespan in culture. They can only survive for so long, so there's only so much you can do with them, and that's not really a good model for eventually going into a cell therapy. So that's why we bother to do this whole process. And my next two questions are, one, how do we produce retinal cells from these stem cells? And secondly, how do we perform gene corrections? So the way our lab has done this is by taking these pluripotent cells and basically mimicking exactly what happens naturally during development of a human. And this is done by exposing those cells to a very specific sequence of proteins called growth factors and other small molecule inhibitors that kind of take the place of different proteins and different pathways. And in this way, we can produce a nice homogeneous population of, these are those pigmented supporting cells I talked about, where they get this beautiful monolayer, kind of looks like a cobblestone patio. And not only can we see that morphologically these cells are what we want, but we can also do things like check for protein expression, which is marked in green here, showing that these cells are actually expressing proteins that are supposed to be expressed in these cells in your eye. And that importantly, they're not expressing any stem cell markers, because of course, if we ever want to use this as a therapy, we don't ever want to be putting stem cells into a patient directly. And then also that they're expressing proteins that are specific to this part of the eye and not some other part of the eye. And this next part I'll blow up bigger on the next slide so that you can see it a bit better. But we can also do functional assays on these cells. So the way this works, this first panel, panel C, is something called a phagocytosis assay. So every morning when you wake up and light hits the back of your eye, those photoreceptors actually shed their outer portions. And the pigmented cells are responsible for binding to that waste, that cellular waste, and digesting it and getting rid of it. So that's a function that's very critical to those pigmented cells, and they have to be able to do that to keep your photoreceptors healthy. And so in lab, as indicated by this gray bar, we can physically feed photoreceptors to our cells, and they will bind them and ingest them and digest them through the same physiological process that's happening in your eye, which is very exciting. And the other functional assay that we do has to do with the barrier properties. So like I said, those pigmented cells are kind of like your skin. And what they need to do is secrete proteins specifically just to the top or bottom side, depending on what they need to be feeding above or below them. And we can show in lab that they're properly secreting certain proteins to the correct sides. And again, this is the take-home message from this data is to show you that if these cells are implanted back into a person, there's a good chance that they're going to function exactly like they're supposed to in the eye. And again, these are some of the cells that are now being used in clinical trials for age-related macular degeneration. And so not only can we make one cell type at a time, but in collaboration with some researchers out of University of Madison, Wisconsin, Dr. David Gam's lab, they've been able to make entire small eyes in a dish. And we call these optic vesicles. They're only 200 to 300 microns, so not full-size eyes. But this is accomplished with the same general principle, where you're mimicking what happens naturally in development to cause these pluripotent stem cells to go through a neural induction and then a retinal differentiation over the course of about 90 days, and then matured beyond that, of course. So this is a cross-section of an eye that we grew in lab from healthy stem cells. And what I'm showing here in red is where those photoreceptor outer segments are. And then in green is a protein that's directly related to that disease, retinitis pigmentosa. So this is sort of a proof of concept that we can, in fact, use these cellular models to directly study retinitis pigmentosa. So I've shown you how we produce retinal cells from stem cells. But what I haven't told you is how we perform those gene corrections, and more importantly, how do we know what we're supposed to be correcting? And so, just as a reminder, I showed you these images of retinitis pigmentosa, of what the patient experiences, what their retina appears like on a large and small scale. But what's still unknown is how this phenotype is arising. What I didn't tell you is that this isn't just one gene that can cause this disease. There's actually dozens of different genes that can have mutations that all lead to this same phenotype. So we really want to know on a molecular and cellular level what is going on. So this was reviewed back in 2006, showing that when they studied lots of families with retinitis pigmentosa, that in some cases it was inherited in a dominant form, meaning one bad copy of a protein will lead to the disease. In other cases it was, in other cases it was recessive, where you need two bad copies of the gene, one from your mom, one from your dad to get the disease, and then also X-linked, meaning it's on your X chromosome. And what's interesting is that all of these proteins are not related. They're not all in one molecular pathway. They're not even all expressed in the same cells. So it's really hard to understand how all of these different mutations are leading to the same phenotype. For example, if you look at just the pie chart for the autosomal dominant form of the disease, about half of the families, unfortunately, the mutation wasn't able to be identified. And then in about a quarter of them, the mutation was in this protein called rhodopsin. And rhodopsin is specifically expressed in your photoreceptor cells, and it's responsible for that process of turning the light signal into a neural signal. And so it makes sense why if you got a mutation in this protein, it would lead to a blinding disease. However, these other proteins that I've highlighted in red are not solely expressed in the eye. They're actually expressed in every cell in your body. Yet for some reason, it's only leading to blindness. So what my thesis work has focused on is looking at just one mutation in one patient in just one of these proteins and trying to nail down what's really going wrong. So again, returning to this idea of pluripotent stem cell disease models, the piece that I've left out is how we accomplish this gene correction. So we know how to make the eye cells, we know where to look for mutations, how are we correcting it? And so you may have heard in the news about CRISPR Cas9. So what is that? Essentially, in the simplest form, they're tiny molecular scissors that can cut out a mutation and splice in the correct sequence. So what does that mean? Well, it stands for clustered regularly interspaced short palindromic repeats, which is completely useless to know, except to know that this is something that occurs naturally in prokaryotic DNA. So this is something that occurs in bacteria. And it's actually the bacteria's immune system. So what happens is when a bacteria is injected with viral DNA, the bacteria can sense that DNA coming into itself. And it produces an RNA, which is a single-stranded form of DNA, that matches some part of the viral DNA. And that RNA can then pair up with this protein called Cas9. And Cas9 is what's called a nuclease, and that's a type of enzyme that can make very precise cuts in the backbone of DNA. So this RNA protein complex binds to where it matches in the viral DNA, and the Cas9 protein makes its cut, and the virus is then disabled. So how did this get translated to human cells? Well, Dr. Jennifer Doudna out of UC Berkeley and her group published in 2013 that not only does this Cas9 protein cut viral DNA, it can also cut eukaryotic DNA, meaning human DNA. And so what you can do with this technology now is if you know where that mutation is in a patient, let's say, and this is their genomic DNA, and you know they have a mutation here that's causing, say, retinitis pigmentosa, you can design that RNA to search through the nucleus and bind to the genomic DNA where the mutation is. And then the Cas9 uses its tiny molecular scissors to cut the backbone of our DNA. And our cells' natural response to damaged DNA is, of course, to rush in and fix it because, as we know, damaged DNA leads to things like cancer. So if you want your cell to repair it correctly, what you can do is give it a little donor piece of DNA with the correct base pair sequence on it. And then it'll integrate that into where the cuts happened, and you now have a fully correct sequence in the actual patient's genome. And it can't really be understated how incredible this has been for the field of medicine, because we have never had the ability to so precisely edit single base pair mutations in human cells like this. And so, like I said, I'm working on this protein called PRPF8. And Dr. Sarah Howden, who was in Dr. Jamie Thompson's lab at University of Madison, Wisconsin, and she's now actually the facility director at a gene editing facility in Australia, published this method back in 2015, where she showed that you could do that simultaneous reprogramming, making those pluripotent cells, and gene correction at the same time. So this first image is just a schematic of the base pair ATCG sequence of the PRPF8 protein. And she's showing her guide RNA in red here that she designed to get the Cas9 to come in here and correct this point mutation that the patient has. And in panel B, she's just showing these sequencing data to show the difference between a wild type person with just a healthy PRPF8 protein and a mutant, and then the result of this correction. And so she has since provided me with stem cells from this patient that are both diseased and corrected, and I'm in the process of producing both those pigmented cells and optic vesicles to really get at what's going on a cellular and molecular level. So to summarize, we have the technology to take a patient's skin cells, produce stem cells, produce retinal cells, and precisely gene edit that patient's cells. Like I said, not only can we make one cell type at a time, but we can actually produce entire small tissues in lab to study several different cell types. And in addition to using skin cells, ourselves and other labs are trying to look at those mononuclear blood cells that have DNA to figure out how to make stem cells from that and go on to make cells for potential cell therapies. And before we can really make this cell therapy a widely used medicine, we're going to need to make sure that we're making the correct cell type for each patient, that we're editing their mutations efficiently, and that there's a way for doctors to safely transplant those cells back into a patient. But if we can figure this out for diseases like retinitis pigmentosa, it will give hope to so many people who suffer from genetically inherited diseases. And I would just like to end by thanking my lab up here on the top. They're a really fun group of people. Pardon my Photoshop skills for getting everyone in the picture. And then, of course, I have to highlight where our building is on the beautiful UC Santa Barbara campus. I'm very lucky to be there working on this project. And of course, I'd like to thank my program at UCSB and all of our funding sources to make this possible. Thank you. Questions?
getting cells that you want? Is that done somewhere else, or do you guys actually have a hand in figuring out how to culture what's required?
Yeah, so the hard labor of that was done by a graduate student in the lab, Dave Buckholz, and he graduated right about the time I was joining. And it was essentially trial and error. So you're basically looking at what happens naturally in development and trying all different timings and concentrations of all these different growth factors that are involved. And he's a little bit of like a god in our lab for figuring this out. So yeah, it was that whole 14-day process of using different growth factors was a lot of reading in the literature and understanding natural development and trial and error, yeah. Yeah, it's a lot of work. Yes.
Yes, I have RP, and so consequently I've done a lot of studying about it. It's my understanding that the rods and the cones are gone, but the connectors between them are still alive, am I correct?
So it depends a lot on those different mutations, unfortunately. So that's what we're trying to get at, because while it's all called the same disease, whether what's happening in individual patients is always the same is still not totally known. Does that make sense? Yeah.
What work has been done, I guess, on what you call the last mile is once you have a set of healthy rods and cones and that optic vesicle, how would you go back and make an eye healthy again?
Yes, so Dr. David Gam, who I mentioned, who's really pioneered the work for making those optic vesicles, another portion of their lab is trying to figure out the best way to surgically implant those cells back into a patient. So with the pigmented cells, it's a bit more straightforward, and that's why it's sort of been the first thing to reach cell therapies and clinical trials, and that's because it grows with this nice monolayer, and we can actually seed that onto a membrane, and the surgeons can go into the back of the eye and create a little blister and place the cells behind the retina and then push the cells back down onto them, which sounds incredibly difficult, and I guess that's why they're surgeons. But for photoreceptors, it's a little bit trickier because you need that correct orientation and you need them to potentially be creating synapses with the other neural cells. And yeah, I won't pretend like that's any small feat, but I think that it is possible, and I think that the better we can get at culturing these cells and growing them fully to maturity and working on those transplantation strategies will make that a reality at some point, yeah. Yeah.
Forgive me, I'm vastly exceeding my knowledge.
Okay.
I know in harvesting and repurposing cells from an existing patient, there's an issue of degradation of the telomeres. And it sounds like that's what you're doing and regressing back to getting the stem cells again. And is that an issue at all? Or?
So I would also be speaking beyond my knowledge a little bit on that. And I think one thing that you may be thinking of, and correct me if I'm wrong, is some of those adult stem cell therapies. So those are treated differently by the FDA in that that's something called minimal manipulation, so where they can take whether it's adipose stem cells from your fat or mesenchymal stem cells or something where they can do some manipulation and reimplant them back into the patient. For this, making those induced pluripotent stem cells, I'm not entirely sure in terms of the age that they were isolated from the patient and how that might affect their telomere length, and if that's something that would be an issue, in the patients. But I don't really know enough about that to answer your question exactly. Yeah.
I'm just curious to know how long roughly do you think it's going to take to get from where you are today to where you can actually change the back of an eye for an RP patient?
Yeah, so for that, with anything that's affecting those pigmented cells, we're in those initial steps. And of course, with any time things go from more of an academic research setting into an actual clinical trial, it maybe slows down a bit in that, so the clinical trial, for instance, right now, anytime you have that phase 1 It's always going to be in very advanced patients where you're doing more of a safety assessment to make sure you don't have any safety concerns. And then in the later stages, you're allowed to go into patients that it might actually rescue some function. So in terms of a timeline, I wouldn't feel comfortable putting an actual number on it. But I do think there's a lot of work to be done to get close to where the photoreceptors are going to be able to be replaced. I think we still...
1 year, it's more like 10 years.
Yes. Yeah. Yeah.
So I have another timeline question, and that is, once you get to that place, what is the time period between when the patient's cells are taken through all the steps to when that patient gets her or his sight back?
Yeah, so that part of the process is another area where there's a few different options. So right now, a lot of clinical trials that are in process are still using embryonic stem cells. So for those, they have cells that have been sort of banked, you call it, where you have them frozen down somewhere and you can bring it to the patient as needed. But there is one patient in Japan that has been actually treated with the induced pluripotent stem cells from himself. And one major issue that I didn't really touch on in terms of making this feasible is to automate this process a little bit. And there's places such as the New York Stem Cell Foundation where they actually have robots that can do this whole process for you. I wish we had $30 million to buy 10 robots, but they can do this whole reprogramming process. And when you take out the issue of human labor to do that process, it cuts down on time and cost. And that's maybe something that would make it more realistic to get it to a point where it's not a year or 2 years to get this whole process for each individual patient. Yeah.
But you made it sound, when you were speaking earlier, that you get it back to the stem cell state, and then you basically let it go forward at embryonic development rates. So you're basically recapitulating, say, the embryonic development of the eye itself. And so it takes about as long as it would to gestate a child.
So that's what we've improved upon. So what I left out is originally we did something called spontaneous differentiation, which is what you're getting at, where spontaneously, just in the time that it takes, these cells would start to appear in our stem cell cultures. And so what that programming is directed differentiation, where we're trying to quicken those initial steps. But for, let's say, the optic vesicles, you can't really make it any faster than those 90 days because, like you said, it takes a certain timing for these things to process. But no, it wouldn't necessarily be 9 months to produce a cell type. There are ways that you can direct it initially rather than going through the whole process of really skipping a few steps to get to that eye state.
Skipping steps is probably active research as well.
Yes, definitely.
90 days is a long time to wait for an experiment to be done.
I know. Yeah, it's good to hear. The good thing they give you 5 years to finish a PhD when one experiment takes 120 days. But yeah, it's a hot area of research, and unfortunately, there is some limit that you reach in terms of how much you can rush the cells. Yeah, but that's definitely an area that a lot of labs are looking into. Yeah.
So I haven't heard of any, but are you aware of any issues with CRISPR-edited genes developing a body response to that, like you would expect from organ transplantation, or has there been basically no effect and they're perfectly safe for the most part that we know of?
So I know in terms of gene therapy, and not necessarily related to CRISPR, but sometimes when a patient is undergoing gene therapy and it's the first time they're expressing that functional protein, they actually can have an immune response to that functional protein because their body's never seen it before. And it's hard to say in terms of the immune rejection. Ideally, these induced pluripotent stem cells would be as close as you could get to the patient's self, yeah. But that's something that unfortunately you wouldn't totally know until it goes through, animal studies and into clinical trials, yeah.
Sure,
question. I'm wondering why the stem cells aren't susceptible to sort of the normal cell division limit for the number of times a cell can divide before it can't divide anymore. Like why can they divide indefinitely?
Yes. In the simplest terms, basically the controls that are in place to terminate mitosis, basically when you get to a post-mitotic state where the cell no longer divides, those controls aren't turned on in these cells. These are cells that are being told to just continually renew themselves. And that has to do with specifically as the cells going through their different growth phases and how certain things are lining up and dividing, both of those cells remain in that stem-like state. Does that kind of answer your question? Yeah. Yes?
You mentioned various proteins. I don't know much about retina pigmentosa. Is the level of proteins that you're picking out, is that consistent throughout the whole process of degeneration of the retina, or does that fluctuate?
Do you mean in terms of which proteins have mutations in them?
You were looking at, you had certain graph with scores around various proteins.
Oh, yeah.
You were looking at. Now, is the concentration of those proteins consistent throughout the whole process of retina pigmentosa? Because when you start out, very few cells are degenerating, as it continues more and more. But is the level in each of those cells consistent throughout the whole process? Versus, say, pick one cell on day one, the level of proteins is the same as day, I don't know, 150.
Yeah. So it would depend on what you're looking at, and the hope is that most things would stay the same, and then you'd find maybe one pathway where things have seemed to go wrong. Maybe something's being overproduced or underproduced, or there's a pathway that's just not communicating correctly, and where things start to go wrong is where you're trying to get your first clue about how this is happening molecularly, and that's what we're sort of in the process of studying, and before you even get to the protein level, we're starting even earlier with looking at RNA sequencing, so trying to get an idea at the RNA level, are there any genes that are up or down regulated at different points as we culture these disease cells out, and that'll give us some hints of where to go from there. Does that make sense? Yeah.
I know this is way blue sky, but are you even close to the geneticists working with the biochemists and the physical chemists to come up with a mathematical computational model of this kind of work so that it can be simulated?
Not precisely that. One thing that we are working with, interestingly, in that realm is a company that does sort of machine learning algorithms where they're trying to train computers to look at our cell cultures and sort of tell which ones are the most efficiently produced. So if we had like a plate where we're trying a bunch of different protein concentrations and a bunch of different protocols, could we get a computer to learn how to recognize what cells are being most efficiently produced? And so in that realm, we are looking down those avenues. It's definitely not my area of expertise as much as I wish I knew how to code a computer to be a machine learning computer. But yeah, it's an interesting area, and I think the program that I'm in, BMSC at UC Santa Barbara, it's a biomolecular science and engineering program, and what's cool is that is what we are all about, is this interdisciplinary interaction between the chemists and the physicists and the engineers and the biologists to get at those questions of how can my techniques and your techniques be combined to get the most efficient results. Yeah, great question. Yeah.
Is there an issue of the cells that you're growing, the stem cells, developing other mutations as that development occurs?
Yes, and so that's a very important point, and that's something that needs to always be considered. And for that reason, especially if anything ever will enter clinical trials, they do a lot of screening for anything that's tumorigenic or stem-like. Unfortunately, recently there were some publications where some embryonic stem cell lines that have been grown in labs have adopted some mutations that can eventually be cancer-causing in combination with other mutations. And again, that's why the FDA has to be so stringent about this, and also why researchers should pay attention to those things. And of course, the reason that kind of happens, as you're hinting at, is as you're growing these cells in lab, you're inevitably going to be selecting a little bit for the ones that are growing the fastest, and that's not intentionally, it's just sort of what happens over time. And so that's definitely a concern, and that's definitely something that always needs to be at the forefront of those conversations, yeah. Other questions? Question? No? Okay. All right, thank you.